Hello. Today we're moving into Chapter 5, which is about the plasma membrane of the cell and the structure and the function of that membrane. If you'll recall from past lectures, we've talked about the membrane actually has a couple of functions that we've already talked about. The first, we said that it isolates the cytoplasm from the outside of the cell. So the membrane is always that which separates the cell from the world. Secondly, we talked about the fact that it is the job of the plasma membrane to regulate exchange of molecules in and out of the cell. So the membrane literally controls what can come into the cell and what can go out of the cell. We've even called the plasma membrane selectively permeable, meaning that some things can pass freely across the membrane and some things can't. So it selects what goes in and what goes out. A couple of other functions that we've not talked about yet that the membrane actually does is it communicates with other cells. There are structures on the membrane responsible for communicating with cells. In addition to that, it also has structures that identify the cell. And keep in mind, every cell has to be identified on several levels. It has to not only be identified as the species of which the cell belongs to. For instance, your cells are identified as Homo sapiens. So there are identifying levels of species. Also, the cell has to know who you are. Each person is individual, so there has to be some identifying mechanism to tell the cell that it belongs to you. And not only that, but what type of cell is it? We call this model, the illustration that you're seeing here, a fluid mosaic model. The reason we call it that is because it looks like a mosaic. If you've ever seen a tile mosaic or a mosaic of tiles, if you look at this membrane from above, it looks like a mosaic. Also, it's very fluid. Predominantly, the membrane is made of phospholipids. Again, these are lipid molecules that are very, very pliable. You should be very familiar with the structure from Chapter 3. Those molecules move around on the side of the membrane where they're located. If we look at the membrane, which this is the illustration, this is what we think the membrane looks like today. And let me also say that that evolves as we learn more about what the membrane looks like. The illustration evolves from years to years. But if we look at all of those structures on the membrane, we could put them into two basic categories. There are two components. All of those could either be classified as a lipid, and by far most of those structures are lipids. We'll talk about the three lipids that are contained on the membrane. Or they could be proteins, and the proteins are responsible for lots of the functions of the membranes. So let's start with the lipids first. First of all, the one that you should be very familiar with from Chapter 3, our study of organic molecules, the phospholipids. And you see here, this is the phospholipid we talked about. There's the hydrophilic head right there, and there's the hydrophobic tail of that molecule. And remember we said in Chapter 3 that those molecules actually form a bilayer. There's two layers of them right there in the presence of water. So you should be very familiar with those molecules and the nature of those molecules and why this membrane is as it is right there. Keep in mind as we talk about things that have to cross the membrane, anything that's crossing the membrane would have to go through that. So you have an area of hydrophilic heads. You have this area right here that is actually insulated from the watery environment of the cell. You have those tails and those tails. So any molecule passing through would have to go through those tails, and then the other hydrophilic heads right here. So as we talk about molecules that pass, think about that. The second lipid is called a glycolipid. It's over here. The outside portion right here is the glyco part. It actually is hydrophilic. These molecules within the membrane serve to protect the cell. It has some protective functions, and in addition to that, it plays a role in cell-to-cell recognition. The third lipid is this squiggly little yellow thing right there. It's called cholesterol. Very important right there. The presence of cholesterol in the membrane actually helps to make it more impermeable. Okay, so let's go to the proteins now. Proteins, these are the globby looking things right there. I think they look a lot like marshmallows in the membrane right there. And if you'll notice, there are some that extend all the way through the membrane, that one and that one. And then there are others that are only on one side of the membrane. We actually refer to those proteins according to how they are oriented. So this one that we saw earlier, this one is just on the surface of the membrane. We call those peripheral proteins. If they are embedded within the membrane, they have a presence on the outside and on the inside of the membrane. We actually call those integral proteins. And notice that this one actually has an opening on both sides of the membrane. So you have this passageway right there, and we'll talk more about that later, but certain molecules can go through that passageway. Some of the functions that these protein molecules might serve, there are some that are involved in cell recognition. We actually call those the cell fingerprints. These are the glycoproteins. And everybody's glycoproteins are different, just like everybody's fingerprints are different. Also, some of these proteins actually are involved, as I illustrated earlier, in the passage of those molecules across the membrane, either from outside to in or inside to out. There are some of those proteins that allow molecules to pass freely, and then others are very selective about which type of molecules can go through them. There are some of those proteins on the membrane that are involved in reactions. A group of them are called receptor proteins. A receptor protein extends beyond the membrane or is on the outside of the membrane, and you can think of those as kind of like gloves out there trying to catch molecules or accepting molecules that come near them or they actually allow for molecule binding. Once the molecule has bound to the protein, it causes the protein to actually change shape, or there's a change within the protein that causes another within the cell. So these receptor proteins allow for molecule binding. The protein actually changes shape and brings about some other change within the cell. There are some proteins that are enzymatic proteins, which basically means that they carry out they carry out your metabolic reactions themselves directly. I'd like to take a look at this little 2-minute video. It's very short, but it does give you a really good idea of the fluid nature of the membrane.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick. Between the living machinery of the inner cell and the harsh conditions of the outside.
So, Moving on, let's talk about what this has to do with you and your cells and the plasma membrane. First of all, we have to look at the nature of fluids and how they react and act. You are very, very fluid. We've said earlier that you are 70 to 90% water. So we really have to understand the physical nature of fluids and how fluids act to understand how things go in and out of the cell. So let's define a fluid as a liquid or a gas. When we talk about fluids, we often talk about the concentration of molecules within that fluid. So concentration is defined as the number of molecules in a given area. We talk about that a lot when we talk about the concentration of things inside the cell versus outside the cell. If there is a difference between two areas, for example, inside the cell or outside the cell, we refer to that as a gradient. So gradient is the physical difference that exists between two areas. So in a fluid, these molecules are in constant motion. If we're talking about the water in your glass or the molecules in the air, it doesn't matter. The molecules are constantly bombarding each other. These collisions are very, very random. But what results from these collisions is a very not random phenomena. If there is a concentration of molecules in a certain area within a fluid, these collisions cause those molecules to be spread out until they are equally distributed. So those molecules that are highly concentrated in one area will move from where they are more highly concentrated to where they are lower concentrated. And this is just a property of fluids. There is no energy exerted here. The movement from high to low is a result of the random collisions. And eventually you'll have a situation that results that's called equilibrium. This is when molecules are spread out equally. Now, once equilibrium is reached, that doesn't mean that the molecules are going to stop colliding. They will continue to collide, and the collisions are random, but things are in equilibrium. What does this have to do with your cells? Well, the cytoplasm inside your cell is very different from the extracellular fluid, or that fluid which is outside your cell. There are lots of concentration gradients that exist. So I want you to think about that for a moment. If a molecule can cross the membrane, it can go through that phospholipid bilayer, and if a gradient exists, and those are two very large ifs, if a gradient exists, those molecules are going to automatically move from the area where they are higher concentrated to the area where they are lower concentrated. And this is done without energy. It's done without any control from the cell. The cell can't stop it. If there is a concentration gradient that exists, they will automatically go from high to low based on that gradient. And again, that's if they can go through that plasma membrane. So molecules move based on gradient if they can cross the membrane. So let's think for a moment about what type of molecules can cross a membrane. What determines if a molecule can cross that membrane? And again, keep in mind, a molecule that's crossing the membrane has to go through a hydrophilic head, two hydrophobic tails, and another hydrophilic head to get across. So things that we have to consider with regard to those molecules would be polarity. You've got to think about, is that molecule charged or not? Because that area of the tails is very hydrophobic. And we learned in chapter 2, I believe, that hydrophobic things don't like charged molecules. So a charged molecule would have a hard time going through two layers of tails. We also have to think about the size of the molecule that's trying to pass. Again, these phospholipids are very, very small. So a large molecule is not going to be able to pass, even if it has the right polarity. Okay, so this type of movement we mentioned doesn't have any energy exertion from the cell. So the cell is not exerting energy for these types of molecules to move. No energy required. And remember, these molecules would be going from high to low. We oftentimes say that is movement with the gradient, moving from high concentration to low concentration. If you want to use an analogy, you could think about it like going down a slide. You're going from a high place to a low place, and it doesn't take any energy to go down a slide. So that's with the gradient. There are actually 3 types of transport that involves this concept. We call them diffusion, facilitated diffusion, and osmosis. So again, all 3 of these are movement across the membrane with the gradient without any energy exerted. And molecules that can do these 3 things would have to have the right polarity, and they would have to have the right size. Again, all 3 of these, no energy required, so we call them passive transport. All 3 of these would be considered transport that is passive across the membrane. So let's take the first one, diffusion. Take a look at that little perfume bottle there and watch the molecules as they leave the bottle. They are diffusing into the air. They are going to from where they are more highly concentrated to where they are lower concentrated. That is diffusion. That's exactly what we've been talking about, diffusion. The movement of molecules from higher to lower concentration until equilibrium is reached. We often say this is down or with the concentration gradient, diffusion. Molecules that can diffuse through the membrane are small, they're non-charged, they're lipid. Examples of those would be oxygen, carbon dioxide, fats, alcohol, and water.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
The second type of passive transport is facilitated diffusion. Now, this is still diffusion, so we're still working under the properties of no energy exerted, and molecules are moving from high concentration to low concentration. But now there is a facilitator. The word facilitate or facilitator means helper. So these are molecules that for some reason can't cross the phospholipid bilayer, so they have to have a helper molecule. And that's one of those proteins that we saw embedded in the membrane there. These are carrier proteins that literally carry molecules across the membrane. Molecules are going from where they are more highly concentrated to where they are more lowly concentrated, lower concentration, but instead of being able to go through the plasma membrane, they have to go through one of these protein molecules. And this illustration represents glucose, which is a molecule that has to go across the membrane through a facilitator or a protein. Charged molecules have difficulty crossing the layer. They're oftentimes moved with a carrier. Molecules that do move across the membrane, like with the carrier proteins, are glucose, sodium, and chloride ions. Any type of ion is going to have a hard time going through that hydrophobic area right here. So they more often go through the membrane there. Third type of passive transport is called osmosis, and this is simply the diffusion of water. A lot of you have seen Osmosis Jones. You grew up with Osmosis Jones, so you should be familiar with the word. We're talking about the diffusion of water. Properties of water are such that when water has a concentration gradient, when there exists a difference in the concentration of water outside versus in, that there is a pressure that builds up. And this is called osmotic pressure. And it's the pressure that builds up because water wants to diffuse, because of osmosis. We show this little example, this little experiment. The concept we're talking about is referred to as tonicity. Very important concept for you folks that are going into any kind of health care. You really need to have a good understanding of tonicity and what the cell is going to do. We're talking about the strength of the solution that a cell is in relative to the inside of the cell. So what are the solute amounts inside and outside the cell, and what exactly is water going to do? Important to keep in mind that tonicity is referring to the strength of the solution. So we're looking at the amount of solutes in the solution compared to the cell. So think about it for a moment. There are three things that could be true in this situation. There could be the same amount of solutes inside and outside the cell, and we call that isotonic. Iso means equal. That's a word you should be familiar with. There could be less solutes outside the cell than inside the cell. We call that hypotonic. Hypo is one of your root words that you should know. It means less. Or there could be more solutes outside the cell than inside the cell. And we call that hypertonic. Hyper means over or more. So let's take a look at each one of these situations and talk about exactly what it is and how the cell is going to react in each of these situations. Let's take isotonic first. Iso, we said, means equal. And remember that prefix iso refers to the amount of solute in the solution outside the cell. So in an isotonic solution, there's the solute concentration is equal inside and outside the cell. So let's take a look at an example of that. And I've tried to simplify it here. I'm illustrating this as being the 5% and the yellow as being the cell, and then the red being the solution outside the cell. If a cell is in an isotonic solution, that means that there is an equal amount of solute inside and outside the cell. So 5% solute, 95% water. What's going to happen here? Water's going to go in and out. Molecules are in random motion. They're colliding. Their molecules are moving. But what is important is there is no net change in the water concentration on either side. The cell neither loses nor gains water in an isotonic solution. If we're looking at an animal cell, water's going to go in, water's going to go out, but there's really no net change in that cell. If we're looking at a plant cell, you have to go back to chapter 4 and think about the plant cell anatomy for a moment. If we're looking at a plant cell, water's going to go in the cell, water's going to go out of the cell, and what's going to happen is this central vacuole, which was the organelle we talked about in plant cells that store the water, the central vacuole is going to fill up with water. This is where the plant cell stores its water. So we get the central vacuole filled up. You can almost think about the central vacuole as like being a little balloon. Excuse me. as like being a little balloon within the cell, and it can fill up with water and it can release the water. And you know what a balloon looks like when it's deflated. So the central vacuole fills up with water in an isotonic solution. This water causes the fact that the central vacuole is full causes the contents of the cell to be pushed against the cell wall. Now this is the cell wall, and remember it's made out of cellulose and it's really rigid. So we've got the contents of the cell. This is the cytoplasm, these are the chloroplasts, there's the nucleus. Everything else, all of this is pushed up against the cell wall. It causes a pressure that we call Turgor pressure, Turgor. Turgor pressure. So when the central vacuole is full of water, the plant cell has lots of Turgor pressure. That's when your plants look really nice and upright. Let's talk about a hypertonic solution. Hyper means over, so we're talking about in a hypertonic solution, we're referring to over, so we're talking about more solutes outside the cell than inside the cell. So in our illustration here, you see we have 20% solute outside, 10% solute inside. So what are we saying here now? 10% solute means how much water right here? 90. 20% solute means how much water? 80. Now remember, the solute's not passing, the water's going in and out. So where is the water going to want to go? We have 80 here, we have 90 here, there's more water here, so water's going to go out of the cell. Easy way to remember is water is going to always move to where the solute is the greatest. Water's going to always follow the solute. Wherever the solute is the greatest, that means that there's less water. So water's going to always go to that area. So water's going to go out of the cell. Very important if you're taking care of people and putting IVs in their arm. You've got to know the cell will shrink. In animal cells, we call it crenation, when an animal cell shrinks because it's been placed in a hypertonic environment. A good analogy. I don't know if you did this as a child or not, or even if your mom maybe did this, but when you find slugs outside, you know what a slug is. It's like a snail without a shell. But oftentimes people will put salt on slugs to kill them. This is exactly what you're doing to the slug. The slug's membrane is water permeable. Water can go in and out. So when you put salt on that slug, you are putting that slug in a hypertonic solution. At that point, there's more solutes outside the cell than inside the cell. So the water from the slug is going to go outside, and you're basically dehydrating them. So animal cells in a hypertonic solution, water leaves the cell. The cell is going to shrink. Again, we call it crenation. In a plant cell, though, let's take a look at that situation. In a plant cell, remember we had this central vacuole right there that was full of water. We had lots of turgor pressure. Well, now the water has left the cell, so the central vacuole has deflated. Now there's no pressure exerting a force for the contents of the cell to be pushed against the cell wall. So what you end up with, we call this plasmolysis, is you end up with the cell membrane, the plasma membrane right there, pulling away from the cell wall, which is right here, and you have all this vacant space right there. You have the loss of turgor pressure. So now what does your plant look like? Your plant is wilted. It won't stand up straight. This is what happens to plants when they are in a hypertonic environment. In a hypotonic solution, hypo means less. And remember, those prefixes are referring to the amount of solute outside of the cell. In a hypotonic solution, there is less solute outside the cell than inside the cell. So let's take a look at our example. Inside the cell here, we have a 20% solute. That means 80 water. Outside the cell, we have a 10% solute. That means 90 water. So remember, solute's not moving. Where is the most water? The most water is outside the cell. So water is going to go from outside to inside where there's more solute and, by the way, less water. So water always goes to the area where the solute is the greatest. What's going to happen? Water's going to go in. The cell is going to swell. If it's an animal cell, this can be very, very dangerous because it can rupture. Very dangerous for animal cells. Think about that for just a moment. If you plug an IV up to somebody's arm that has pure water in it, if it's 100% water, you know that we are 70 to 90% water. So if you put pure water in someone's bloodstream, the red blood cells are going to rupture. They're going to swell. They're going to take all that water in, and eventually they will rupture. So in a hypotonic solution, animal cells, water goes in. The net movement of water is into the cell, and they can rupture. We call that lysis. Lysis is a word you should be very familiar with. This is about the 4th time we've used that root word in the course. So lysis means to burst or break. In a plant cell, though, take a look at what happens to a plant cell in a hypotonic environment. Net movement of water is going to be in. The central vacuole is going to get really, really full. You're going to have lots and lots of turgor pressure, but plant cells don't rupture. They don't lysis. And the reason for that is because they have this rigid cell wall right there that's containing the cell. So plant cells will not lysis.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Some molecules can't cross the membrane passively, and they have to be taken through actively. So the cell literally has to exert energy to bring them in or to remove them from the cell. It requires energy on the part of the cell, and we oftentimes see this occurs when molecules are moving against the gradient. Let's talk about what against the gradient actually means. If we said with the gradient means from high to low, like going down a slide without energy, against the gradient would be from low to high. So you're taking molecules from where they are least concentrated and moving them against the gradient to where they are more concentrated. So from low to high. Again, our analogy of the slide. If you're going from low to high, you're going to have to use some energy to get there. So we're moving against the gradient. This is done two ways in the cell. The cell can use carrier proteins. These are those integral proteins, some of them that are on both sides of the membrane with the passageway, or sometimes vesicle transfer, vesicle mediated movement. Carrier proteins, again, we said had an opening on both sides of the membrane. Energy is required. These are pumps that act as, these are proteins that act as pumps. They literally pump molecules across the membrane. Your muscles do this. Anytime we're moving an ion, oftentimes it has to be pumped across the membrane. Anytime something's going against the gradient, it has to be pumped across the membrane. A great example of some molecules in your body that go against the gradient have to be pumped are iodine molecules that have to be moved into the thyroid. Your thyroid is an organ that has to have iodine to function. Yet any iodine that comes into your body in the bloodstream needs to go to the thyroid. So the thyroid is concentrating iodine in that organ, but yet it still wants all of the iodine that you bring in. So it's having to pump those iodine molecules from your bloodstream, where they are least concentrated, into the organ itself, where they are more concentrated. So the molecules are going against the gradient. Vessel transport is also another way to bring large molecules into the cell or release them from the cell. This is when you have molecules that can't go through the membrane and they can't go through a protein, but they need to come in or out of the cell. And they do so by vesicle formation. Going into the cell is called endocytosis, and we actually saw this in the endomembrane system in Chapter 4. We saw molecules being brought into the cell by vesicle formation. You see here that these molecules, if this is outside the cell and this is inside the cell, these molecules are coming into the cell. The plasma membrane is forming a vesicle, and now we have the molecules enclosed in that vesicle right there coming in. It would be something like that, very simply put. And think of it in some of these one-celled protists that we're going to talk about later in the course. They actually use endocytosis to bring in their food particles. What you see here is an amoeba, and it's actually taking this particle in by endocytosis. It's forming a vesicle around it. Exocytosis is releasing substances by vesicle formation. And you have the vesicle inside the cell that has molecules in it. Excuse me. Let's go back. You have the vesicle right here inside the cell. Having some technical difficulties. You have the vesicle there inside the cell that literally is fusing with the plasma membrane and releasing the contents of that vesicle outside the cell. Notice that the membranes that were included in the vesicle have now been recycled and are present in the plasma membrane. What's outside the plasma membrane of plant cells? We've already talked about this. Plant cells have walls outside the plasma membrane. Let's go ahead and make it clear that whenever a cell has a wall, and we've talked about several cells that have walls. We've talked about fungal cells that have walls made out of chitin. We've talked about plant cells that have walls made out of cellulose. We even talked about bacterial cells that also have a cell wall. Anytime a cell wall is present, all molecules are freely permeable through the cell wall. Everything can go through inside, outside the cell wall. The cell wall never controls passage. It's always the plasma membrane's job to regulate the entrance and exit of molecules into and out of the cell. So the cell wall of plant cells, freely permeable, and it's made of cellulose that we talked about in Chapter 3. Outside of an animal cell, down here, this is inside the cell and this is outside the cell. Outside the cell here, we have something that we call the extracellular matrix. Extra means outside, so we're outside the cell. And if you look at some of the words there, you can see that it's really busy. It's a meshwork of lots of different types of molecules, but they are all polysaccharides and proteins there. So this kind of helps keep the cells together, this meshwork of polysaccharides and proteins. So what really is holding cells together? Let's take a look at plant cells and some of the junctions between plant and animal cells. Plant cells first, okay? The junction between plant cells, if you take a look at that cell, you'll notice that between the cells, there's openings in the cell wall here between the cell. Those little openings right there actually have cytoplasm, strands of cytoplasm that are going from one plant cell to the other there. Those are called plasmodesmata. Those are strands of cytoplasm that link adjacent plant cells. So every plant cell would have plasmodesmata with the plant cells next to it, helping to hold them together. As far as animal cells, there's several different types of junctions between animal cells, and the junction between the cells depends on what type of tissue we're talking about. What type of cell is it? Some cells need to be held together very tightly because they're under some undue stress or force causing them to stretch. Other animal cells are not under such stress, so they just need to be held together just so they don't come apart, but they're not under any type of stress. Let's look at the first one right here. Those are called desmosomes. You would find desmosomes between perhaps skin cells. These are called adhesive junctions. They're more like spot welds. If you see the strands there between the two cells, those filaments there, just every once in a while, if you were sewing, this would be like tacking two pieces of material together. They're not under any type of force or they're not stretching. So, and if it's your skin cells, you're going to slough them off anyway. So, they're just held together, with those strands just intermittently there. Some cells, like the example down here in the corner, some cells need to be held tightly together because they're under some type of force or stretching. Those are held together by tight junctions. These are zipper-like. You've got those adjacent proteins between the two plasma membranes bonding them together. If you were sewing, this would be like zigzag stitching two cells together. They are never coming apart, ever. You would find these type of tight junctions perhaps between cells in your bladder, the bladder tissue, as the bladder is stretched when it fills up and it's under all that pressure from being stretched. Those cells are never coming apart, and it would be devastating if they did, because you would end up with urine in your abdominal cavity, which would not be good. The third type is called gap junctions. These are little donut-shaped structures, and they have a passageway that allows for molecules to go between cells. These are found in cells that need to communicate instantaneously, cells that need to always be in communication. For instance, heart cells have gap junctions. Very important that all heart cells communicate because they all have to contract at the same time. If they didn't contract at the same time, then you would not have that property of pumping that's so important to that organ.